DNA fingerprinting introduction. DNA fingerprinting is a technique used to identify individuals based on their unique DNA profiles. This method is grounded in the principle that no two individuals except identical twins share the same DNA sequence. It has application in forensics, paternity testing, and genetic research. DNA fingerprinting was first developed in 1984 by British scientist Sir Alex Jeffrey. The technique revol- revolutionized forensic science by proving a reliable method for identifying suspects. Over the years, advancement in technology have improved the accuracy and efficiency of DNA analysis. Now, basic principles of DNA. DNA is composed of nucleotides, which include a sugar phosphate group and a nitrogenous base. The sequence of these nucleotides encodes genetic information unique to each individual. Variations in DNA sequences, such as STRs, are key to DNA fingerprinting. Sample collection. DNA can be collected from various sources, including blood, saliva, hair, and skin cells. Proper collection and preservation of samples are crucial for accurate analysis. Forensic investigators often use swabs, tubes, and other sterile tools to prevent contamination. Extraction of DNA. DNA extractions involves breaking down cell membranes to release the DNA contained within. Chemical reagents are used to separate DNA from proteins and other cellular materials. The quality and purity of extracted DNA significantly affect the results of subsequent analysis. PCR amplification, polymerase chain reaction. It is a technique used to amplify specific DNA sequences. PCR allows scientists to create millions of copies of particular DNA segment for analysis. This, this step is essential, especially when the initial sample is limited or degraded. Analyzing DNA profiles. After amplification, DNA profiles are generated using such techniques or gel electrophoresis. This process separates DNA fragments based on their size, creating a unique banding pattern for each individual. The resulting profiles can be compared to determine matches or exclusions in forensic cases. Applications. It is widely used in criminal investigations to identify suspects and victims. Can provide conclusive evidence in court cases, often leading to exoneration or conviction. The technique has also aided in solving cold conclusive evidence in court cases, often leading to exoneration or conviction. The technique has also aided in solving cold cases and identifying unidentified remains. Paternity testing. DNA fingerprinting is commonly used to establish biological relationships such as paternity. By comparing the DNA profiles of a child and potential parents, one can determine biological connections. This application is crucial in legal cases involving child support, custody, and inheritance. Ethical considerations. The use of DNA fingerprinting raises ethical questions regarding privacy and consent. There is a concern about the potential misuse of genetic data and the implications for personal privacy. Ongoing discussions aim to balance benefit of DNA technology with the protection of individual rights.